One more stop on our way to understanding how Punnett squares work, and we're going to look at another specific type of inheritance. And in this case, we're using blood types. Blood types are the only example of multiple alleles that you need to be able to understand. There are others out there, but multiple alleles means specifically that one gene, up until now, even with sex linkage, each gene has only had two forms, a dominant or a recessive form. So we talked about eye color. We talked about color blindness. You have an X big C, that's for normal vision, and an X little c. So there's only been two variants, two different alleles. So multiple alleles, we're talking about three or more alleles for the same gene. And it turns out that blood types are an example. Blood types are an example of multiple alleles because we have three different forms. And again, I'll use a question to illustrate this. And so we'll go through and then try to learn about it as we're going through. So the way that the IB wants you to understand how to represent these alleles is to use the letter I, and a capital I means it's dominant. There's another capital I, and a lowercase I means it's recessive. So that follows the same rules: dominant, recessive, big letters and little letters. But in this case, we have two dominant alleles, two dominant forms. They are IA and IB. And IA and IB are co-dominant to each other. What that means is when they're both present, if I get an IA from my mother and an IB from my father, that I will express both of them and I'll be a special phenotype of AB type blood. So they're co-dominant, which means they both affect the phenotype when they are actually present together. So same way that we've tried every single question up until now, we need to establish what these letters are and what they mean. IA is the allele for blood type A, IB is the allele for blood type B, and little I is the allele for blood type O. So now let's approach this question. A man with blood type AB marries a woman with blood type O. Show the possible blood types of their children. Now, in this case, although we have man and woman that are stated here, I know that blood types and multiple alleles are not sex-linked. You do not have to, and as far as I'm aware of, there are not any specific X-linked traits that have multiple alleles that you would have to study at this particular level. So in this case, I don't need to include X and Y chromosomes. Remember, if it's color blindness or hemophilia, those are the two probably most famous sex-linked traits, so you will have to include X and Y chromosomes. But blood types, you don't have to. So again, the most difficult two parts are establishing what the phenotypes and the genotypes are of the two parents. Then the rest is cake. So let's start with the parent phenotypes. A man, one parent is blood type AB. So what's his physical trait? Okay, it's just blood type AB. That's easy. The other parent is blood type O. Nothing tricky about this question. Physical trait, blood type O for the other parent. Now I have to figure out the genotypes. Well, I know each parent can only have two alleles. That's a rule. You can't have three alleles for the same gene. So this blood type AB parent, got an allele from mom and an allele from their dad. Same with this parent over here. So this one's not too hard to figure out. If they're blood type AB, then that means they must have inherited an IA from one parent and an IB from another parent. So here we go. An IA from one parent and an IB. That's it. I can't get two. They can't have three parents making one baby. So that's it. Blood type O, well, let me review here. Blood type O is caused by the allele little I, and that's recessive. So this has to be like this, a little I and another little I. If there was an IB in here, well, this wouldn't be blood type O because B is dominant, so this would be blood type B. So this blood type O parent has to have two little I's. Let me double check that. That's okay. IA, IB, blood type O can only be this. That's it. Fantastic. Now you just separate out the gametes, the possible gametes. So once you get used to this, you can skip a few of these stages here and just go straight to the Punnett square. But I like writing it out for consistency, and it helps me make sure I don't make any mistakes. So there we go. There are the two possible alleles. There are the two possible alleles. Up until now, I've put all the gametes in a circle. It just helps me to understand that those are the actual gametes. You can do that. You don't have to do that. But now we move on to our Punnett Square. Again, the parent, the man, or the father doesn't really, the man or the woman doesn't really matter. But I'll just keep it consistent. So the man is AB. So this is the man, so AB. So his possible gametes, if I'm just going to move these down here. Oh, I have extras here. So IA moves here, and IB over to here. The woman, the mother, is blood type O, so she can give a little I or a little I. Okay, that's fine. You may realize at the end of this that when you have a situation like this, you don't necessarily need to do the entire Punnett Square, but this is just a monohybrid cross, so it's not too hard to do it like this. But you'll see, if I can remember later, after I fill in these boxes, if I just ignore this row down here and only fill out these two boxes, the ratios are still going to be the same. So that's a quicker way to solve these if you want. So carrying on, what do? Bring these down. I can bring the little I over and then an IA. Now remember, we always want to keep things consistent, keep things neat, keeps housekeeping. So all the dominant ones will go first. It means the same thing, but this, again, in the previous video I said it just looks like you don't know what you're doing. By convention, we just always put the dominant one first if there is a dominant and a recessive there. Does that make sense? Okay. This IB and an I will come together. Here we have it, nice and convenient, ready to go. Good. Here we have same thing, IA and a little I, and I have it all ready to go right here. Just drag it over. So just the same thing, IB, little I, same thing over here. So what's my conclusion? Well, I like to put in the phenotypes. So what do I have here? So always add some space so you can put the phenotypes, and then you can go back and look what the question is actually asking. So in this case, this is definitely going to be blood type A, because that IA is dominant. This is definitely going to be blood type B, this is definitely going to be blood type B, and this is going to be blood type A. So in the end here, what do I have? See, notice if I actually summarize this, 50% of them will have blood type B and 50% will have blood type A. If I covered this entire row down here and just didn't bother doing this because it's the same, the ratio is still the same. It still tells me 50% of the kids will be A, 50% of the kids will be blood type B. So I have a 1-to-1 ratio of type A to type B, and I can also say that all the kids will actually be heterozygous. So each one of these kids, when they grow up to have their own kids one day, they could still pass on the little I, and they could end up with kids of blood type O or blood type A or B or even AB, depending on who their partner is. One thing important to keep in mind is if this question in the beginning said, man with blood type AB marries a woman with blood type A, and that's all the information that's been given, well, someone with blood type A could have two possible genotypes. They could be IA or IA little I. So if they don't give you all that information, then you have to do two separate Punnett squares to figure out the possibilities, and then you'll have a ratio out of 8 possible outcomes. That the percentages might be a little bit different. So watch out for that. If you have blood type B or blood type A with no extra information, know that there are two possible genotypes there if they don't tell you if they're homozygous dominant or heterozygous. So you might have to do two Punnett squares and then do the total ratios from there. Okay, I think that's it. So in this, using blood types, we've learned that blood types are an example of multiple alleles where you have more than two forms, 3 or more alleles for the same gene. And we've also learned the word codominant, where codominance could be in flowers. For example, you could have a red allele and a white allele that happen to be codominant with each other, and if they're codominant, then that means you'll have a new phenotype. So the flowers might even be pink or something like that. So you have to know if the trait is codominant. The ones that you will have to know from heart are blood types for multiple alleles, color blindness and hemophilia for sex linkage, and then anything else they give you, they'll tell you what it is. So if they give you something strange like, I don't know, the disease PKU is inherited in a recessive fashion, they'll tell you that how it's inherited, and they'll tell you if it's sex linked or not, and then you'll know whether there are 3 alleles, or if you need to include the X and Y chromosomes, or if you just do a standard monohybrid cross. Okay, I hope that was helpful. Have a nice day and enjoy doing many genetic practice questions. They are very fun.